Hello, friends. Hello. How's everyone doing? Welcome back from the coffee break. I hope we're all refreshed. I hope we're all appropriately caffeinated for some of the very interesting technical talks that are going to come next from New Harvest Fellows. I really love that this session is following the last session because we're kind of connecting the dots here, right? So we got on stage with Jake and we talked a lot about open source technologies and open research and development. And this is one of the main strengths of New Harvest, and where New Harvest's role in this community really shines through, is through their support of these fellows who are doing foundational research that they are publishing openly that is advancing the entire field along with them. These fellows are going to go on to found companies. In fact, some of them have already kind of started. They're going to go on to be the thought leaders and technical leaders in the cellular agriculture space as the field scales. So I'm very, very excited to bring them up on stage to talk to you a little bit about their research that has been supported by New Harvest through this fellowship. So up first is going to be Ricardo Gouveia. Yes. Dr. Ricardo Gouveia, who is a fellow from Newcastle University. Stephanie Kawecki, who is at the University of California, Los Angeles, and Sam Peabody, who is working out of Texas Tech. And after this series of lightning talks, there's going to be a panel moderated by Dr. Breanna Duffy. Bree is the research director at New Harvest and a tissue engineering scientist herself. So give everyone a big round of applause before they come up, and Ricardo is going to come to you.
Thank you so much, and thank you, New Harvest, for this amazing opportunity to present for you today. I forgot my click. Where is the clicker? So, my name is Ricardo Gouveia. I'm a postdoc fellow, very kindly sponsored by New Harvest. Thank you very much. And my talk to you today will be entitled Form and Substance, addressing the challenges to produce structured meat at scale. Of course, at this point, we are doing proof of concept stuff, but it's important to start somewhere. But first, let's address this elephant in the room. So, full disclosure, I'm a research associate at Newcastle University and will be presenting today work undergone as a research fellow for New Harvest, but I'm also the co-founder and CSO of a startup company out of Newcastle University called 3D Biotissues. And my initial interest was biomedical engineering. So I was very much focused on developing new processes and products that could be applied to cure diseases, in particular those addressing structured tissues, such as cornea, skin, and other tissues that are very prone to disease via the loss of form and/or substance. And this resulted in a relatively successful body of work involving understanding a bit more about stem cell biology, serum-free medium to sustain and grow these cells, as well as create new solutions for advanced tissue manufacture. And together, this also allowed me to develop a number of skills and expertise that are very intersectional, from molecular and cell biology all the way to materials and material sciences and biomaterials, that ultimately are incredibly important to understand how the biology of tissues work in order to better replicate the biosystem in vitro, and use this as an iterative system to deliver products and processes for multiple applications, such as clinical or industrial. At the time, I was not looking into food, but that is something that New Harvest ultimately allowed me to understand. is that the approaches that I was taking for tissue engineering substitutes for human transplantation, that is, the ability to control cell growth and produce large quantities of structured tissues, the development of serum-free medium solutions to grow these tissues, and the accurately reproduce the tissue structure and composition, that is, the function of the tissue, could easily be applied for the engineering of tissue substitutes for human nutrition instead. Applying exactly the same principles, but in this case, not only the structure and composition that we want to obtain in order to reproduce function, is also to reproduce taste. My project was mostly divided into main streaks, if you will. One that was developing new biochemical factors that would increase production yields of these tissues using elements or new ingredients that were sustainable and chemically defined. That is something that was very important and is very important for GMP manufacture, for clinical applications, and of course sits at the front and center of culture and meat research. And in particular, I was interested in addressing the role of macromolecules and the effects of macromolecules in the development of such biochemical solutions. The other was, of course, using very simple biophysical cues. In this case, substrate geometry to better control how cells align and reproduce their tissues similar to what they do in vivo. And as you can see in some of these images, it was quite successful to do this in a fairly small scale. You can see a comparison there between the natural skeletal muscle fascicle and a culture muscle tissue that we were able to produce in small amounts that ultimately resulted in the production of something that has a certain structure to it. And that has been published and is being published as we speak. Regarding the biophysical cues, we'll talk a bit more about that later, but for the macromolecular crowding effect, one of the important aspects is that it was fairly successful, but not entirely so. Again, this is an iterative process, and having a certain degree of success is already a very good, promising start. In particular, I want to highlight the bars in orange. These are the conditions on which C2C12 myoblasts or mouse pre-adipocytes were subjected to macromolecular crowding effects. And as you can see, they significantly improve cell proliferation in serum-free medium conditions compared to serum-free medium without supplementation. However, they are still not as good as serum-supplemented conditions. But when you look at the amount of tissue that these cells are able to produce, then you see something remarkably different, because they are also producing extracellular matrix, and that contributes to the total biomass production. And in fact, these macromolecular crowding effects are mostly seen in a massive increase in extracellular matrix production. So in terms of tissue biomass, we, at least for muscle cells, we obtain increase over FBS supplemented conditions. And the same can be said regarding conditions where you co-culture muscle cells and fat cells in the same surface. So very similar results for cell proliferation compared to FBS, and improvement on biomass biofabrication. Now, I told you that we also were comparing the ways the cells and the tissues were being formed when exposed to biophysical cues such as milliscale curvature. And what we observed is that the cells behave very differently and counterintuitively when they are grown on surfaces that have a curvature that is about 7.5 to 20 millimeters in diameter. So this is much bigger than the cells, several orders of magnitude larger than the cells, and it's surprising that the cells are responding to such big geometric cues. But they do so. They do so, and they align perpendicular to the long axis of the curvature. And you can see the differences here between structured tissue and non-structured tissue, where the cells, in this case mouse myoblasts, are much more aligned compared to cells that are growing on flat surfaces, and that also leads to increases in cell density, tissue density, and biomass production, so final biomass production. We are talking about a very simple geometric cue. It's not active. You don't need to have a high precision of biofabrication. You just need to have certain curvatures. Now, this is incredibly important. Why? Because we also demonstrate that structured tissues have also different elastic or tensile properties compared to less structured tissues. In reality, when you look between the comparison of structured versus non-structured, you see that tissues that are grown on curved surfaces have tensile properties that are much closer to mouse muscle tissue from the leg compared with adipose tissues and non-structured materials. And this is also important because these tensile properties affect how these tissues are digested. This is an in vivo digestibility model that we performed in collaboration with another startup from Newcastle University, where you can see that the digestibility profile of our cultured meat is different independently if it's structured or non-structured, and is definitely distinct from products like mycoprotein or just ham that you can buy on the supermarket. So again, a lot of things that have been addressed, but what I want to highlight is that there's not one elephant in the room, there's an herd of elephants in the room. I hope I could convince you that we were addressing some, or made some advances in addressing some of these elephants, but there are many more to do. And also convince you that the form and substance are important, interdependent things. Finally, I just want to leave you with a provocation, which is very intentional. Many of us do form versus substance. So this we're talking about the bull in the room. And things like, oh no, we alone can do it. No one else does it better. What we know, and this experience, or this new harvest experience, let's call it, has shown us, is that this industrial academic synergies really accelerates research, reduces wasteful resources, and promotes accountability as well. The other thing that you typically hear, especially when you're talking to media, is, oh, we'll have it on the market next year or in 5 years, soon, sometime. Look, it took decades of open source research, thousands and thousands of scientists and medical clinicians, billions of dollars in public investment for us to deliver a very successful COVID-19 vaccine program worldwide that is arguably orders of magnitude simpler to do compared to what we want to do in cultured meat. And finally, well, some hype is good. I think no hype is even better because good products and efficient processes tend to speak for themselves. So thank you, everyone, for your attendance and your, thank you.
Hi, everyone. So I just wanted to start off by saying that a little almost 5 years ago, I attended my first conference in cellular agriculture, and I was a volunteer at New Harvest 2017. And I remember seeing the fellows present their research and immediately thinking, that's what I want to do. And so I'm just really excited to be here and want to thank you all for having me and thank New Harvest for the opportunity. So today, I'm going to talk about one of my primary research projects, which is working towards building multi-component tissue in 3-dimension, and using these composite and tunable scaffolds so that we can cater to each cell type. And right now, I'm primarily focusing on engineering muscle tissue and adipose tissue. Again, my name's Stephanie, and I am at the Rowett Lab at UCLA. So early on in my program, I had the opportunity to collaborate with behavioral scientists to begin identifying what are some of the barriers that cultured meat are facing in terms of consumer perceptions. And what was interesting is out of all of these top barriers, taste emerged as one of the top concerns. And so I found this opportunity to use my background in science and engineering to begin addressing this barrier. So then I started thinking, well, what makes meat taste really good? And doing a deep dive into the literature, it's really well established in the field that fat is really important for meat texture and flavor. In particular, intermuscular fat contains delicious lipophobic flavor molecules that consumers crave. And in fact, all around the world, we use the level of intramuscular fat as a grading system to determine the quality of our cuts of steak. So after doing this important research and researching and getting these findings, and many late nights, I derived an equation to come to some conclusions about my findings, which is fat equals pretty hot and tempting. And it's really what consumers crave. And yet at the time when I started looking at this, not a lot of projects were focusing on engineering adipose tissue. And even in the biomedical industry, compared to other tissue types, it's something that there's just less literature on. So I decided I want to focus on engineering fat, and I want to do it within intramuscular or within muscle bundles in 3-dimension, and I want it to be thick and pretty much make a steak. And I think if I could go back and tell myself the exact challenge that I was getting myself into, I might have, give myself a little bit of a warning. But in all seriousness, having the opportunity to work in academia and be funded by New Harvest really set me up to kind of take on a more high-risk project and a long-term project. So then I started thinking, how do we make steaks in vitro? One approach certainly is 3D printing. And in fact, last year we had an amazing proof of concept done by Aleph Farms in collaboration with the Israel Institute for Technology. And while this is amazing strides and we're making great progress in this field, and it's certainly a promising approach, but we're going to need more innovations in technology to bring this to scale. Plus, I feel that it's really important to have redundancies in our food system and develop other processes that reach the same goal, especially if there's any issues with gaining materials or anything with infrastructure issues. It's good to just have redundancies and so that we can always deliver on our products. And so to simplify this approach, I started focusing on first muscle and fat and began considering what are the types of things that I need to take into account as I'm trying to engineer these tissues in 3-dimension. And first, one of the considerations is the topographical patterning. So it's well known that muscle cells that are grown on aligned substrates differentiate more efficiently, and it drives their formation of multinucleated myotubes. There's also the consideration of mechanical environment, and both cell types have been shown to grow and differentiate more efficiently on substrates of physiologically relevant stiffness. And so for muscle, it's a little bit stiffer, and fat, softer. They also have very different media requirements, particularly when it comes to differentiation. And then spatial orientation. So if we're trying to do this in one pot, it's challenging to direct the cells where to go to form the intramuscular marbling. And of course, diffusion limitations. So if we're trying to make thicker cuts, In the absence of vasculature, we're really limited to about 1 millimeter in thickness, which is quite thin. And so here, with all of these considerations, I'm trying to think, how can we address this? So I developed a modular and tunable scaffold approach to address these considerations. And so here, I'm able to fabricate scaffolds that cater to each cell type. So for muscle cells, I'm fabricating aligned gelatin nanofibers that are a little bit more stiff, and I can able to seed my cells onto that and grow them separately so they could drive and form myotubes. Similarly, I can grow adipocytes on these softer edible gelatin microbeads, which have been shown to result in higher lipid accumulation within adipocytes. I can then grow each cell type on their respective scaffold and media formulations as well. And then I can use a bottom-up approach to build three-dimensional tissue with differentiated, two different differentiated tissue types, and also have these adhere to one another on the time scale of hours. So first, I wanted to make sure that the cells were doing well on their respective scaffolds. And so as a first step, I seeded C2C12 mouse skeletal muscle cells onto these aligned gelatin nanofibers. And what I found is that they were aligning and differentiating, as evident through the red myosin heavy chain staining. I then wanted to quantify their myogenic index, so that's the number of nuclei within a myotube divided by the total number of nuclei with the sample. And I also wanted to see how this translated across the different cell types. So I also looked at primary rabbit skeletal muscle cells. And what I found with both of my samples is that growing these cells on an aligned substrate resulted in a higher myogenic index. I wanted to continue quantifying and confirming this, so I performed quantitative RT-PCR, looking at 3 muscle differentiation markers, and I compared unaligned fibers with aligned fibers and also tissue culture plastic. And while there was not any statistical difference in the expression between unaligned and aligned nanofibers, for all 3 samples, there was statistically higher expression of these markers compared to tissue culture plastic when grown on an aligned scaffold. So next, I wanted to confirm how my fat cells were doing. So I seeded 3T3 mouse pre-adipocytes on the softer and edible gelatin microbeads, and I stained for lipid tox to visualize their lipid accumulation. So I saw that they're doing well accumulating lipids. However, I wanted to quantify this, again, using RT-PCR and looking at 2 fat differentiation primers. And what I found is that there was no statistical difference in terms of expression when growing these cells on the beads versus tissue culture plastic. Although it is important to note that growing the cells on these microcarriers is a more scalable approach towards engineering adipose tissue. So what do these cells look like post-stacking? So here I'm taking a confocal image sort of at the edge of my sample so that you're able to visualize some of the muscle layer as well. And I've stained again for myosin heavy chain, so it indicates that we have differentiated muscle. And I also immunostain for PPR gamma, which indicates that adipocytes have differentiated, as well as the lipid tox staining. And so we can see that we have both differentiated cell types onto a composite material. And I think the most surprising part about this is that they were actually able to self-adhere to one another. So initially in my proposal, I put that I was going to add an additional cross-linker like transglutaminase, but I found that I did not need to do that, which was really interesting, and it's a fairly stable structure that results in this process. But next, I was wanting to see how this translated to a primary cell type. So I did the same approach with rabbit skeletal muscle cells and rabbit pre-adipocytes. And what I found is similarly, the structures were able to adhere to one another, although what was interesting is that the primary cells take a lot longer to adhere to one another. So while I'm not entirely sure if it's scaffolds interacting with one another or cells and scaffolds or cell-cell, this result kind of indicates that cells may be playing a role in adhering to one another, and maybe we can build these 3-dimensional tissues without additional cross-linkers, which would help with the elimination of a post-processing step. Another thing that was interesting is by fats, as you can notice, there's not as much a lipid accumulation with the rabbit cells. And at first I was like, what's happening? Am I doing something wrong? But then I thought rabbits are quite lean animals, and so maybe naturally the cells just don't accumulate as many lipids. And then I thought how interesting that we're entering a world where we could have a marbled rabbit steak, which doesn't normally exist in nature. So I'm just really excited to see kind of what culinary opportunities and food experiences that we can bring into this next food revolution, which is really exciting. So then I wanted to look at the mechanical properties. The graph looks a little sad, but those 3 points actually took a lot of work because I had to upscale the tissue to be about 25 millimeters in diameter and about 3 millimeters thick. And so that was just a lot of scaffolds and culturing, and I was able to make this upscaled version and use rheometry to calculate the Young's or the elastic modulus to determine how stiff the mini meat is. And what I found, it's actually ranging between 2 and 3, so a little bit on the softer side. If you look at tissue, it's a little bit closer to fat at this point in terms of texture. Certainly some things to improve this would be adding connective tissue, also any post-dehydration steps. I think one thing that I haven't seen a lot of research on is what are we going to do after we harvest the meat, and looking at that science between transforming a carcass to meat and changes in water content and pH, which is something that I'm very interested in. But all of these things are going to play a role on the ultimate mechanical properties of our products. So in conclusion, I'm able to use this approach to make aligned gelatin scaffolds that can be catered to muscle cells to differentiate, as well as using a different type of scaffold to differentiate fat cells. And then I can build these composite materials using myogenic and adipogenic microtissues that are able to adhere to one another on the time scale of hours without the use of additional additives. So hopefully that's could be a positive kind of thing in terms of processing in the future. I'd love to thank my lab and my PI, Amy Roat at UCLA, all of our funders, and of course, New Harvest, who I would not be here today if it wasn't for them, and be as far as I am in my cell ag career because of them, so thank you. Oh, it's gone. It's okay.
Alrighty. We good? There we are. Afternoon, y'all. My name is Sam Peabody. I'm going to be talking about food safety in cell culture foods, microbial modeling and detection. And I just want to preface this first by saying that I'm a food scientist. I'm not a tissue engineer. That's sort of my wheelhouse. And what I want to do is just have y'all zoom in real close, and then we're going to kind of zoom out a little bit on how operations in a cell-cultured food establishment might work. And then from there, we're going to just sort of discuss how microbes happen in there. So we're going to start with a concept called the 6-log problem, although maybe 6-log challenge is a better term for it. It has to do with the concept in food science, which is the fact that our limit of detection is, by convention, around 1 CFU per mil. In principle, you could hypothetically find 1 CFU per liter, but that you would need a very large auger plate to plate that 1 liter, which is not very practical. That being said, what are some characteristics of the 6-log problem? The fact of the matter is, very large bioreactors may be more prone or more susceptible to contamination. That has already been discussed, I think, a couple times here today, and it's certainly part of the reality. The other part of this aspect is that the level of contamination might not be observable because of that aforementioned limit of detection until it's too late to do anything about that. And that can be a serious challenge, especially if you're in a food safety environment where you're wanting to ensure that whatever is growing in there is not a potential risk or hazard to your customers. So some of the research I've done at New Harvest with support from New Harvest has been to model Staphylococcus aureus in DEMEM with 5% FBS. I've also modeled Listeria monocytogenes and Salmonella enterica species, strains. And in this particular model, it's Staphylococcus aureus at 37 degrees Celsius. And we see very quickly it overtakes the media, going to a very high concentration. So it would seem that it's very much so indeed. And that's not entirely unknown. I mean, that's pretty obvious. Everyone here who's had tissue has probably encountered contamination at some point. But we do need to think about, like, what is the model for pathogens specifically? And just going a little bit more into the causes of the 6-log problem, one concept that's really important to understand, and this is something of a distinction between cell-cultured foods versus, say, a brewery or somewhere in pharma, is the lack of what we call in food science competitive inhibition. I believe Ken talked about it a little bit more, how the presence of microorganisms on food can actually inhibit pathogens from occurring. We simply don't have an immune system operating within a bioreactor. It just doesn't exist yet. There's been some interesting suggestions on what could be done there, but I'm not going to focus on that today. But I do want to put out that idea that we might want to consider this more like a pasteurized dairy product rather than like a pharma output, because that's really the kind of modality I'm thinking. Some other things to bring up, again, I think these were covered a little bit earlier, is that stainless steel that's used in bioreactors and other equipment can fail. It can be susceptible to weakening and degradation over time, especially in the presence of high-pressure steel. That can create small niches within the production process that are opportunities for microorganisms of any kind, both pathogens and non-pathogenic, to spread and accumulate over time. This is, in fact, also an ongoing challenge in the dairy industry, where they do have to routinely ensure that their tanks are sanitary for operation. But we can do a lot about that. And one of the things that we can do about that is that we can be mindful of what's happening in our establishment at all times and everywhere. And I don't just mean within the bioreactor itself, where you might take a look at the consumption of oxygen or of a particular carbohydrate or what have you, but also within the establishment, like the areas around it. You want to be mindful of what the map of your establishment looks like, because that can help you establish what susceptibility you have to contaminating or possibly recontaminating your bioreactors. Other opportunities you can take are to optimize your processes. As I said before, there is a distinct growth curve rate for Staphylococcus aureus and all these other organisms. Depending on how quickly and capable you are of optimizing your bioprocess at the late stage, you could be able to get what you need to get done and then get out before the accumulation of any contaminants becomes an issue. And that's something that will have to be explored and evaluated on the company-by-company basis as bioprocesses are innovative. But it's certainly worth discussing, and it's certainly worth understanding. And last but not least, of course, eliminating unnecessary risks. That means just engineering out all those susceptibilities and vulnerabilities, because if you don't have the risk in the first place, it can't be a problem. But I do want to go back to that top one, environmental monitoring. Because in our research group, we call it enviromapping as well, so you may hear me go back and forth, so forgive me for that. But in environmental monitoring, what we are looking at is we're looking at what is the footprint, what is the signature and indication of microbes within an establishment. And it doesn't just have to be a cell-cultured food establishment, it's by and large the practice in establishments everywhere. And the reason that we have environmental monitoring is because it is a verification process that we use in either a food safety plan or in a HACCP plan. It is a verification process that ensures that we have compliance with regulators. So by having an enviromapping system in your establishment, you are saying, hey, FDA, USDA, not only are we making this happen, we're proving how we are making this happen. So you have sanitary controls in place. You have all those kinds of good stuff. And a couple other thoughts about EMP, because I have a little limited time, so I can't go into all the details, but please come to me afterwards. I'd love to discuss it in more detail with you, is that we have this concept of hygienic zoning, where we look at how close and how much proximity certain surfaces and materials and equipment have to the vulnerable areas of our process, and look at, and using a targeted approach, determine what kind of susceptibility or vulnerability is on it. Obviously, surfaces that are closer in proximity or are more likely to be a cause of cross-contamination need to be explored more, hence why I say targeted approach. You need to be active and assertive in looking at that. And one other thing that's worth mentioning is the concept of microbiome study. This is when you take all the organisms that you collect from your enviromapping and you look at the whole genome sequencing with a shotgun spread. So you're not looking at a specific organism, but you're just looking at what genes are present in the samples that you do collect. This can give you insights into what kind of genes are present at the beginning of your process, and then what kinds of genes are present at the end, so that you know what are the indications of which spoilage organisms might occur. And while, yes, the inside of the bioreactor may very well be sterile, you do want to consider whether or not there's something inside your establishment that's falling on it after it gets exposed to air, because at some point, presumably, you have a conventional food processing point. So it's very important to have that in consideration. And especially, as I said before, I did Listeria monocytogenes cultures. When we're talking about environmental mapping, we definitely want to look for Listeria, because if you have a ready-to-eat product, and I think there's more than one establishment out there in cell-cultured foods that's looking to do ready-to-eat, then you absolutely want to make sure that you're controlling for Listeria, because it is one of the biggest ones we look at when we're talking about ready-to-eat products. And if you have any questions about it, I'm happy to talk about it. The USDA has a whole 80-page document discussing that. It's a long read. But yes, moving on from that. So why have these? I think I've already made the case, but I'm going to really nail it in there. Food safety mitigates losses. If you have a recall, you have to make the voluntary choice of pulling your product off the shelf. It's a choice you and your company have to make to spend money. It's very expensive. Also, the USDA and the FDA are not rubber stamps. They're not just like a quick, easy approval. They are scientists. They are people that look at the facts and the data out there. And if you make mistakes, you're creating facts that they then have to learn from. And they will learn for you if you do not learn for them. That's just a part of the reality. And this is a very important point, because this is about controlling the narrative. Who's assuring to consumers that these products are safe? Who's assuring that these products are fit to consume? Y'all are. If you're in Cell Ag, you're the ones that's creating the story. You're the ones that have control over the story. And right now, it's cheaper now to market and promote these products than it will be when there's any kind of event that causes an outbreak. Classic examples we have, case studies we have from the food industry in general, include incidents like flour. Flour used to be considered pretty safe until we found E. coli in it, and now it's no longer considered safe to eat without some kind of thermal process. I believe another classic example is peanut butter. Peanut butter, in certain contexts, can be of a matter of concern, whereas before it wasn't. There's many case studies out there, and I'm happy to discuss the particulars of those case studies as we go forward with this discussion. That being said, controlling that narrative is really important, and we've encountered some people who were eager to work on that narrative with us. We were able to obtain some samples from WILDTYPE, in which we consulted with them and looked at killing salmonella in their product. We chose, at Texas Tech University, the temperatures to work with, and gave them some valuable data that could be insightful for them as they provide recommendations for their consumers on the heating profiles that would work with their product. We choose these temperatures because, as you can see from the data, we go from taking about 20 minutes to kill salmonella to less than 2 minutes. And the reason we choose salmonella specifically is because among the foodborne pathogens that we take into consideration, salmonella tends to be the one that's more resilient to heating. So if you can kill salmonella, you can reasonably expect to kill other foodborne pathogens as well. And with that, I just want to throw a couple more thoughts out there on this, which is that building partnerships is really key to a thriving cellular agriculture ecosystem. That means like getting that food safety data out there and making it accessible, because having that kind of openness and accessibility not only informs regulators, but it also informs consumers, and it creates a sense of transparency and trust. And you always have that until you don't. And so it's very important to be proactive on that, to prevent rather than to try to deal with the problem after the fact. Again, a marketing campaign that's promoting your product is a lot more effective than a marketing campaign that's apologizing. It's just a reality. So while intellectual property is a motivator for profit, and by all means, have your processes, have your secrets, have that stuff, that's good. Get an advantage, be competitive. But we also need to take into consideration, what do we need to share with each other and among each other so that we can be advantageous for each other? Because it's not so much an issue where if one company screws up, that company's gone. One company can inadvertently damage the reputation of many companies. And that's why working as a collective is so important. as I said before with those other case studies like flour, it wasn't all the companies that suddenly had E. coli in their flour. It was one company. And then it became an issue for all companies. So keep that in mind. And so with that, I would just like to wrap this up by saying thanks to New Harvest. Their initiatives put me up there. They gave me the opportunity to really think about food safety and cell-cultured foods, and I wouldn't be able to relay that and share that information to you without their support. I want to say thanks to Texas Tech University and the International Center of Food Industry Excellence. That is a mouthful. They've been very supportive to me throughout the years, allowing me to go to my first conference in 2018, and every year that there's been a conference since then, we've brought more people, and I hope to just continue the conversation and be supportive, not only to individual players in the industry, but the industry as a whole as I can. And special thanks to WILDTYPE for being just awesome friends who have been continuing the conversation on food safety. We had the opportunity to share with them our generic food safety plan that we created about a year back, and we're happy to share that generic food safety plan that we have with y'all at any time in the future, if you want to just get an idea of what that looks like. So that's all I have. Thank you very much.
She can take it.
All right, hello everyone. I am Duffy, and I am one of the New Harvest Research Directors. And in that role, I have the pleasure of supporting all of our incredible fellows that are doing research like this around the world. So really excited to get into some questions with them after those wonderful presentations. So I just want to start off with a question for all of you, which is that even now, cultured meat research in academia is still pretty rare. And when the three of you started, it was really rare. You were really some of the first people to do this type of research in academia. And I think in every case, the first ones in your lab to do cultured meat research. So I'd just love to hear from each of you a little bit about what that experience was like to really be on the cutting edge of this.
Well, it was really, for me, it was a huge privilege to see that something that I was very much not involved in could be so thoroughly explored. So whenever I was doing research in biomedicine, the idea was always try to see look for a plan B, to try to see how else can this be used for, and how can this be applied. So to have something that is not immediate correlated was a huge privilege and fantastic. More recently, I've been trying to push forward cultured meat research within my university, and at PI level and students level, graduates and above, the reception is quite good. Then ultimately, there is a lot to face beyond just the initial interest in order to make it more established.
Yeah, definitely makes sense. Stephanie?
It's definitely been really exciting. And just being able to track the progress of the field through the time in my program, it's just growing exponentially. And being there at the beginning, though, you know, it had its challenges. At times, it was lonely being the only person, not only in my lab, but in my program to be working on cell-cultured meat. And I did get laughed at a couple times by people, but the majority of the reactions were people are so positive about it and interested and can actually relate to it. And I think that was my first experience when comparing to my other projects, especially where it's like, oh, I'm doing this, and people are like, cool. And then when I talked about cultured meat, people actually had some opinions about it because people could relate to it. So I felt like I was in the right place because I've always wanted to be in a project that was translatable and that people could relate to. And so while there was, challenges, even like pre-New Harvest, felt lonely. After New Harvest, I felt like I had a great team and, just knew that I was in the right place. Yeah.
Yeah, getting started in a CellAg, applying for a New Harvest Fellowship, getting that all set up, I think in 2019. That was interesting. First off, when I went to my first conference in 2018, I didn't know I was actually going to get approval when I asked, but I had an amazing professor at the time, Dr. Mindy Bashirs, who was very supportive and encouraged me to go, and that really pushed me forward. And I have an even more supportive professor, a very much so supportive professor now, Dr. Marco Sanchez. They gave me a lot of support while I live in a state where brisket on the biscuit is the norm. And so I do know a place where we can get that. Just ask me later if you're ever in Lubbock. But yeah, there was definitely some uphill battles with feeling confident in what I was doing and questioning whether or not I was in the right space, because for a long time it seemed like maybe it was just a passing fad or something like that. But the reality is no, it's a very serious work. And with my understanding of global food security that I gained from learning under Dr. Marco Sanchez, I see how we have new opportunities and avenues for solving the questions of hunger in different parts of the world. Maybe not necessarily directly, but by understanding the holism of food production systems and changing the way we produce food, we can address those as well. And that, to me, is very important to it. And even if there are some folks around Texas that look at me kind of funny when I talk about this stuff, I'm not bothered by it anymore, because I know there's an even bigger community of people out there who want to innovate and create it and build a new future.
Yeah, so cool to hear. So I can't help myself from asking, but I would love it if a few of you could, or each of you could talk a little bit about what it was like to be doing cultured meat research before and after the New Harvest Fellowship.
Well, in terms of personal experience, or?
Yeah, from your personal experience.
I think being one of the first really drives home that you are building the initial critical mass. So there's also a bit of responsibility there, that your research will start not just advancing the field, but actually building the field from scratch, or at least doing your best to do so. And that is ultimately what us as academics are known for. It's the curiosity, isn't it? Trying to be as much curious as possible, and trying to challenge preconceived ideas and try to address that with our best skills and capacity. So I feel that ultimately it is very rewarding for us to see the field grow because of our direct contribution, surely.
Yes, that's so critical, and we'll get to that idea a little bit later.
Yeah, if I could comment. I think the biggest difference was speed of research progress, and starting in a lab that didn't have designated protocols to the work that I was doing, and going from that to figuring everything out by myself, essentially, to having this large network with so many resources and people doing similar work, and just the open access information and sharing of protocols, things that I was starting off new. I didn't have a background in bioengineering. So things that, relatively, might be considered basic or simple protocols, but it takes a while to really get, a good protocol down, and also, get the experience needed to be successful at it. And really having that resource is so just invaluable, it's just priceless, really, and it helped so much.
Big changes for me before I took on a research project with support for New Harvest, and after. When I was first learning about cellular agriculture in the context of cultured meat, my head was a bit in the clouds on it. I thought of it as like a passing fancy, a cool idea, and I had tons of ideas. I even tried to see if there was a possibility of patenting one at one point. I was so ambitious in my thoughts. But then I applied for a New Harvest Fellowship and got a lot of grounding, and got really humbled by the amount of thoughts and brilliance of all the other fellows I encountered over the years. And it's been very impactful for me, because it's taught me how to listen more and think more, and really just know that the way you bottle lightning is with collaboration, because that lightning is in the minds and hearts of all the people that you interact with. And coming out of a New Harvest project, I see a bright future with a lot of potential, but it also requires that interconnectivity. And despite things like COVID and all that, which at times seem to make things more difficult, I feel in some ways it's also brought things closer together because those of us who are pushing forward are really connected to our mission and goal, if not substantially so now more than ever.
Yes, definitely. And I want to circle back to this idea of open research that we've been talking about all day, and that critical mass of research we need to come out of academia. And each of you have kind of dealt with that in different ways. Sam, I'm interested to hear a little more from you about the challenges of doing food safety research when so many companies are very secretive about the products they're doing, and how we can encourage more of that collaboration.
So the way I look at that is, while yes, the processes and intellectual property of every corporation or institution out there is its own, there are some things that simply can't stay hidden. And when it comes to regulation, there's going to be certain levels of compliance. Now, obviously, when we're talking bioprocess, there's a lot that goes into that. It comes from pharma that really, that's sort of the mode of thinking. But it's not a food science type of thinking, and it's not taking into consideration what food scientists do, because the profit margins in food production are substantially lower than what we would see in pharma. So there's a completely different mindset for how to look at bioprocess. And while individual processes do need to be protected, it's really important also to try to integrate concepts like FSMA and HACCP into those bioprocess designs as they become food for human beings. And I think that answers your question. But yeah.
Well, and we're all happy to see the collaboration with WILDTYPE. It's so exciting to see the work that's going to come out of that. And Ricardo, you have also had that experience with doing open research and founding a company. I'd love to hear a little bit of your experience of kind of how that works, and obviously how the importance of open research plays into that.
I might say that these are very different mind frames and mindscapes when you're talking about doing open research at the academic level and then working at a company. Because ultimately, again, for researchers and academia, the driver is the challenge and the curiosity. There's the driver for companies, deliverables. You have very well-defined timelines, you have product pipelines, and you need to achieve those. So, and these two mind frames are sometimes not very compatible. So ultimately, there's a lot of juggling and adaptation when you're migrating from academia to industry in that regard. It is feasible, but again, it is a hard transition. Now, in terms of how can we find better synergies within this with these two mind frames, I think we heard from Sam and earlier today that there are things that do not need to, they are not very good to be secretive. There are things that don't really impact the business plan of the company, so they might very well be published and disseminated as much as possible. And then there's the point where many companies ultimately are doing things that everyone else is doing and repeating and wasting time and resources that are so precious, basically falling into the same traps. Whereas if this was published and available open, not only you would not do that mistake and repeat others' mistakes, but you'd allow others to further explore different avenues of inquiry that you cannot, because you don't have the resources or don't have the time or even the idea to do so. And those can then contribute to your own business. The solutions that others create might be very useful for you. So ultimately, I think there is a balance to be had there.
Yes, definitely. And that balance is hard to find, but it's good to see it happening.
No, yes. No and yes.
And Stephanie, do you have any thoughts on challenges you saw throughout your degree in trying to keep things open and work in pre-competitive spaces?
Yeah, I think we've had little success building a collaboration with a company, and it's always initially an exciting, meeting and ideas, and everyone gets really hyped up, and then all of the kind of legal aspects come along, and then it just kind of, falls to the wayside, unfortunately. So I don't have as much experience in that sort of capacity. I would love to. But yeah, and I think that's probably the main issue is that I haven't, been able to experience that because it's, all of the legal aspects and different kind of goals in terms of research and.
Yeah, and that itself is valuable information showing where those barriers are, and it is so important that we learn how to break those down. So your experience is really helpful. And I can't see how much time we have left. Okay. So I guess just generally, do you guys have any thoughts on ways that we could increase this ability to bring people together on pre-collaborative or pre-competitive collaborative work? Any final thoughts?
Yeah, I do actually. I think I was trying to get at this before and didn't quite finish the point on it, but as I was saying before, of course, protect bioprocess design if you must. But also look at some of the downstream operations that are not vulnerable to IP theft or patenting. Look at some of those downstream processes that are normalized in food science and food safety. If that's something that is already out there and you can disseminate information on that and make that publicly available, by all means, please do it. That's an easy throwaway. Don't take the money that you spend on safety and hide it from the world when you can use it as an indication to say that, hey, this is us being transparent. Hey, this is us. Get the most use out of your research if you can. And if you can turn that into a little bit of marketing, by all means, do it. Don't spend your dollars. Try to spend your dollars twice if you can.
That's good advice. Okay, and then I think we're going to take some audience questions. Are we still doing that? Nope. Okay, it looks like we are out of time. All right, thank you so much for sharing your research and chatting a bit about what it's like to be in academia.
Thank you.